Everyone, I'm Zhimin, come from Fujian Agriculture and Forestry University of China. This is my presentation. In Arabidopsis, the female genital phyte development includes mixed progenesis and mixed genital. During mixed progenesis, the MMC specialization is a sign of the initiation of female gene cells. The present studies classify these genes regulating the MMC specialization into groups. One group of the genes regulate the formation of MMC, such as SPL. Another type of genes regulate the number of MMC, such as TAX1-28. The ovules are embedded in the floral organ, so it is difficult to observe the ovule development. Fortunately, some studies have linked the floral organ development to the ovules development. For example, at stage 10 of the floral organ development, the ovules is in MMC stage, and some researchers used the unsequencing of ovules at MMC stage to find KLU, which regulates the number of MMC, but traditional unsequencing analysis used the tissue block RNA, which masks the heterogeneity of cells. But single cell unsequencing can overcome this defect. In recent years, the high-throughput technology of single-cell unsequencing has widely used in plant study, such as root cells in Arabidopsis and rice. So we want to use this technology to explore the female germline deprivation in Arabidopsis. According to the observation, the process of MMC specialization can be divided into AC, MMC1, MMC2 stages. To obtain single-cell expression profiles of ovules primordial, we collected the protoplast of ovules at this 3 stage. The basic information of single-cell unsequencing shows in the table 1. Then we performed the graph-based clustering of each sample and get 8, 10, and 10 cell clusters. Then we examined the transcriptomics communication and the expression level of venous single and the MMC SMARC genes. And we found that they are enriched in cluster 1 in AC stage, cluster 10 in MMC1 stage, cluster 6 in MMC2 stage. So we defined these 3 clusters, AC.1, MMC1.10, MMC2.6 as the female germline associated cell clusters. But these 3 cell clusters account for 5% to 16% of total cells, which is more than half the percentage of MMC. So we generate the sub-epidermal cells and got 5, 4, and 4 subclusters based on the transcriptomics communication and the expression level of venous single and MMC SMARC genes. We found the subcluster 1 in AC.1, subcluster 3 in MMC1.10, subcluster 3 in MMC2.6 can be seen as the female germline-related subclusters. And the result is also confirmed by heat map of the top 20 figure genes of these 3 cell clusters, and also confirmed by the transcriptomics communication of other MMC SMARC genes. And we also found some new genes which may play a role in the MMC stabilization, such as PGDH1. Then we use the SPL-P1, which are expressed in the epidermal cells to identify the sub-epidermal cells. Based on this gene's expression level and the transcriptomics communication, we found the epidermal cells, subcluster 1 and 2 of MMC1.10, subcluster 1 and 4 of MMC2.6. WOK28 is specifically expressed in the epidermal cells surrounding the MMC using these genes. We found the subcluster 4 of MMC1.10, subcluster 2 and 4 of MMC2.6 can be seen as the epidermal cells surrounding the MMC. Then we reconstructed the MMC differentiation trajectory, and the result shows in figures A and B. The distribution of cells along the trajectory shows that the female germline-associated cell cluster, the cotton some intermediate cells, and then we mapped the non-MMC SMARC genes, DMC1, ASL1, SDS, AG9 onto the development trajectory, and we found that this bridge is about a germline bridge. Because F shows the dynamic repartition of the genes along the trajectory, and in germline bridge, the genes play a role in chromosome organization, cell cycle, and so on. We also found there are 200%. We also found that there are 200% 5TFs in DGs along the trajectory, and their extreme potential extreme waves suggesting an intra-strict regulation of female Gemini differentiation. PGDH1 and 8040-13710 also completed the bigger genes of the female Gemini associated cell clusters. So we examined their expression pattern. PGDH1 expressed in the epidermic nucellus near the inner integument from AC stage to MC2 stage. Another gene is mainly expressed in the central design of the nucellus, and this extreme pattern suggests that the superphytic cells surrounding the MMC may close the molecular figure with the female Gemini, and the frequent molecular exchange may occur between these cells. ERL1 is a bigger gene of the epidermal cells, so we generate the extreme pattern of the erecta gene family, and we found they are expressed in the epidermal cells from MC1 stage to post-mitotic stage, which imply the erecta gene family play a redundant role in the MNC specialization. So we generate the triple mutant of the erecta gene family, and we found that the mutant have two defects: multiple MMC-like cells formed and the MMC counter differentiation. And this result suggests that the erecta gene family play an important role in restricting MMC specification to a single cell and promoting the MMC differentiation. This is a summary of my study, and our study indicates the single cell unsequencing is a pivotal in identifying female Gemini differentiation, and it also provides reference for the study of germ cell development of other species in plants. That's all. Thanks for your attention.
Thank you. This is a very complicated study. Thank you for sharing it with us.
Thank you.
Yeah, we still have 2 to 3 minutes for